Let's talk about euchromatin and heterochromatin. See, there's a basic difference between euchromatin and heterochromatin. Chromatin is a term which means colored thread-like structures. They are actually stainable materials that are present in the nucleus of the cell. Chromatins are made of DNA, histone and non-histone proteins. In prokaryotic cells, we don't find any chromatin because there's no histone and non-histone in prokaryotic cells. Because of the presence of basic amino acids like histidine, arginine and lysine in them, they are positively charged, and this positive charge facilitates their binding to the DNA because DNA is negatively charged. Now, the histones are of 5 types: the H1, H2A, H2B, H3 and H4. The amount of DNA and the amount of histone are equal to each other. The amino acid sequence of histones are evolutionarily conserved even in distantly related species. It is very simple to understand how much important the histones are for our existence. Now the non-histones are the other types of proteins that are associated with the DNA, but they are not that much abundant. Non-histones are acidic in nature, which means they are negatively charged instead of positively charged like the histones. Histones are evolutionarily conserved, but non-histones can vary. Even in the same cell types, you can see different types of non-histones. Now, the histones H2A, H2B, H3 and H4 form histone octamers, around which 147 base pair long DNA is wound about 1.65 times. The H1 binds to the middle of the DNA segment wrapped around the core complex and to the linker DNA. Now, the linker DNA is the strand of DNA which connects two individual nucleosomes. So, nucleosomes are the structural unit of the eukaryotic chromatin. Now, let's come to the main topic: euchromatin and heterochromatin. Euchromatins are the regions of the chromosomes that become darkly stained in the metaphase stage and lightly stained in the interphase. Most of the genes are active in euchromatin, which means they produce proteins or functional RNAs. So condensation and decondensation in cyclical pattern occurs. Euchromatin does not have repetitive sequences which can be found in heterochromatins. They are more darkly stained than the euchromatins, both in interphase and in metaphase. The heterochromatins can be of 2 types: the constitutive heterochromatin and the facultative heterochromatin. Constitutive heterochromatin is always silenced, but facultative heterochromatin can be expressed when needed. Hope this video was helpful. Thank you very much. Have a great day.